Okay, so I will now speak to you about the tools in Bgee. And so first, the tools which allow us to access the expression codes. So there we have a gene page. We've already shown it a bit to you. You've seen it in several of our slides. The gene page for each gene which has data in Bgee, you will see the expression conditions defined by anatomy, development, sex, or strain. It's all integrated per data. It's all integrated, but you can also select to see only what comes from, say, bulk RNA-Seq or single cell RNA-Seq or RNA-Seq bulk and single cell or in situ and so on. We give both the significant present or significant absent calls. So do we have evidence that this is significantly expressed above background noise, or is it absent? And this both from direct observation from propagation. So if it's present in the cerebellum, it's present in the brain. We provide orthologs and paralogs from the database OMA of orthology. The paralogs are only in-species paralogs. So if you have a human gene, you find a human paralog, which can allow you to go to expression comparison. And we provide cross-references to UniProt, Ensemble, and so on. So we also provide the present absent calls not in the gene, outside the gene page, but in these downloads. So you can go to our website and download. It's moving again. I don't know. You can select our. Something is wrong with my PowerPoint today, and I don't know what it is. Sorry about this. I'm sorry. I'm going to restart PowerPoint because something is really weird. Okay, I hope it will stop changing. And so here you see you can select conditions in the same way what Frédéric showed you, but here you can select conditions to download files which tell you whether genes are present or absent. So if you want to find, you're working on, say, cerebellum, and you want to filter your data by genes only expressed in the cerebellum, only want to see interactions between genes expressed in the cerebellum, only want to see SNPs of genes expressed in the cerebellum, or anything like that. You can download these files and import them into R or any other environment, Python, what you want, and use these present absent calls. So I'll go to WooClap now. So a little question and sorry. So Bgee highlights annotation of genes to the gene ontology, to expression anatomy, to expression disease, or to expression development. What is highlighted in Bgee? Do you see the WooClap? I don't have the green border around it. Hmm?
Yes, we see.
Okay. Okay, so most of you replied expression anatomy. I think we've been speaking a lot about anatomy so far. So yes, that's what we highlight. Although we do annotate to development and sex and strain, what we highlight is the anatomy. The first thing you see on the gene page by default is the anatomy. The first thing by default you download present absent files is the. We do have gene ontology terms that we take from Ensemble, but this is not what we highlight, and we do not have expression disease since we only have wild type healthy. Now we have another tool which is a bit more original, a bit different from what you've been seeing so far. So when I spoke about ontologies at the beginning of the course, I said, for example, there's the gene ontology, and it can be used, for example, for gene ontology enrichment. So I think most of you have done this. You get a list of genes which corresponding from the output of a GWAS or differential gene expression, or a scan for SNPs, or a scan for selection, or the genes involved in a disease or anything. And the first thing you're going to do with this gene list is what on earth do they do? You'll do a gene ontology enrichment. But you can also learn about the properties of this gene list in other ways, by same logic of looking at enrichment. And so we propose this tool called TOPANAT. So TOPANAT is going to do the same as the gene ontology enrichment, but instead of saying, do I have an overrepresentation of some gene ontology terms associated to my genes? I'm going to say, do I have an enrichment in some anatomy terms where my genes are expressed? So it's based on the fact that we have this curated wild type healthy data, so that it will give you the expression not dysregulated in tumors, for example, or in mutants. And it's based on the fact that we have a reliable, robust call of presence of expression to associate this gene is expressed in this anatomical structure. So every gene has expression in anatomical structures, and some structures are represented many times in the list. You get the list of genes, maybe only one of them is expressed in the hippocampus, but many in the cerebellum. So for example, here I have a gene list, which comes from a review on genes involved in autism and another neurodivergence, which I forgot. And these genes, we have 46 of them, which are expressed in the frontal pole. And so we can look, and we find that 46 are expressed in the frontal pole in this list, and 56 are not expressed in the frontal pole. And if I look at the other genes in the genome of the human, 3,400 are expressed in the frontal pole, and 32,000 not in the frontal pole. So by chance, if these genes were drawn at random from all the genome, I would expect 6 times less genes here expressed in the frontal pole in proportion. So I have a 6-time I would expect 6. So I would expect 6. So I have a 7.6 time enrichment. 46 is 7.6 times more than the 6. This is highly significant by a statistical test, hypogeometric test. And so we have way more of our genes involved in autism which are expressed in the frontal pole than expected by chance if I took from the whole genome. And so you can do this for every anatomical structure in the same way that for a goal enrichment you do for each gene ontology term. For each anatomical structure, which genes in your gene list are expressed there, which are not expressed there, and on the background of other genes which are expressed, which are not expressed. And because the ontology is a graph, you have non-independence. So if genes are expressed in the cerebellum, they're expressed in the brain, they're expressed in the nervous system. So you don't want to test the same thing several times. The same applies to the gene ontology. If something is a hydrolase, it's an enzyme. So there have been statistical methods developed to correct for this non-independence of the terms in the graph. And so we have modified a tool called TopGo, which does what is called deconvolution of the graph. You can read the original paper if you're interested in the methods, but for our purposes here, what's important is that the same exact mathematics applies to the Uberon anatomy ontology. And so we apply exactly, we have modified TopGo, that's why it's called TopAnat, like TopGo, but for anatomy, so that you can apply exactly the same tool. So by default, because it goes faster, we do not do a deconvolution of the graph, but on our website, you can, or on the R package, you can do all the tools of deconvolution which are in TopGo. And it gives you information on the properties of the genes. So here, for example, I went to Flybase, the model organism database for Drosophila melanogaster, and I asked for all the genes where when there's a knockout or a knockdown, you have a phenotype which affects fertility. So you have reduced fertility. And what do I find? I'll make my pointer more visible. There. I find that the genes are highly enriched in expression in cells or associated to reproduction, which you expect. So oocyte-associated follicle, nurse follicle cell, germline cell. This makes a lot of sense. A lot of genes are expressed in the reproductive system. If you mutate them, you will get infertility or reduced fertility. But you also get neuron, for example. And why do you get neuron? Because a lot of the mutants which reduce fertility in flies have to do with behavior. So if the males don't recognize the females anymore, or if the females don't recognize the courtship of the males, things like that, you have a lot of behavioral mutations in flies which will lead to, in practice, reduced fertility because they do not mate. And so these genes which give reduced fertility in fly, you can see that they broadly fall into expressed in the reproductive system or expressed in the nervous system. And here I cut the list to enter the screen, but actually it goes down and it continues in these different types. And you have a link here, which when I will share the slides after the course, you click, you can see this analysis. It's shareable and reproducible. Now, if you do any such enrichment analysis, whether it's our TOPANAT or any gene ontology enrichment, be careful of the pitfalls. And the main pitfall is background. So when you compare, if you go back to how these tests are done, it's this contingency table, right? You have your gene list and you compare it to an expectation, a null expectation you have from this background. So if you change this background, you change your expectation, you change the results. So here, for example, I compare it to all the genes in the genome. But suppose I was not able to study all the genes in the genome. So suppose that I have orthologs between several species, and I'm comparing those which duplicated to those who did not duplicate. Then my background should only be the genes for which I can find orthologs. The other genes cannot be studied, but the set you cannot study is always biased in terms of gene ontology and in terms of expression. If I have used, for example, here there's all the genes of the human genome, coding and non-coding. That's why you have such a big number. If I used a method which only analyzes coding genes, if I did a DNDS, if I did a GWAS on exon capture, I did an exon capture where I only see coding genes because of my protocol. If I started from an RNA-Seq with poly-A capture, then my background should be only the protein coding genes. Otherwise, I'll have a bias. So it's very, very important when you do such an enrichment that your background corresponds to the set which you could test. Now the problem is they have multiple testing because I said we do this on every term in Ubron, and if you do a gene ontology enrichment, you do it on every gene ontology term. So you have many, many terms you test, tens of thousands, which is why in TOPANAT we always provide both a p-value and an FDR correction, post-discovery rate correction. And you have non-independence of terms. As I said, if it's enriched in cerebellum, it will be enriched in brain. And so for this, we have algorithms in TopGo, which allow you to correct for this. And the two main ones are ELIM, which will allow you to find the most specific terms in priority, or WAIT, which will find the more general terms. TOPANAT, yes?
But can you query for senescence? So TOPANAT is really about retrieving anatomical terms. You can ask to retrieve anatomical terms only in some specific developmental stage. By default, on the web interface, I think it's only embryo and post-embryo. You can refine for other developmental stage from the BGDB R package. Otherwise, you could also perform an enrichment on the developmental stage because it's also an ontology. We don't provide that on the web interface or the R package, so you would need to download our data and perform the enrichment yourself. But so, short answer, you can subset the developmental stage you're interested in to retrieve the analytical terms.
Yeah. And this syncs well with this, thank you, Frédéric, with this slide. So on the web, you can easily use TOPANAT. So you want to use it, you just paste your gene list basically and go ahead. It's very easy. Warning, it's a bit slow because the calculation with the deconvolution, everything takes time and it's running on our server and we're not the NCBI, so be a bit patient. Typically takes half an hour, or you can run it locally in the R package. Then the speed will depend on your computational resources, obviously, but especially you can be much more fine. So you can say, I only want expression in humans above the age of 65, for example. We could do separately. I want to know how it differs in senescence or in youth. So I will do one analysis with expression in people between, say, the age of 25 and 45, and another between 65 and 85, and another above 85. And you can do this very finely in the package where all the information is available. We spoke a lot about comparing between species this morning. And so to compare, you have to compare what's comparable. You probably all have heard of autologous genes. These are the genes which are comparable between species. But if we want to compare expression, we need not only to compare genes which are comparable, but to compare patterns of expression which are comparable. What does it mean to say this gene has the same expression in a human and a zebrafish? You need to know what is comparable. And the criterion for comparison, whether it's for genes or anatomy, is homology. So in Bgee, our curators also spent a lot of time annotating manually which organs or substructures anatomical are homologous between species. And this is from primary literature, it's from old textbooks, paleontology, evolutionary developmental biology, zoology. It is not just from our expression. We don't just cluster our expression, say if it's the same expression, it's the same thing, because it can happen differently. It can be that there's convergence of gene expression on things which are not homologous, or it can be that there are structures which are homologous but have different functions nowadays. So this entirely comes from evidence from paleontology, developmental biology, and zoology. And here, for example, I took all the tissues, all the organs which were studied in GTEC and human, and asked what are the homologs in zebrafish. So you paste here these identifiers, you ask between these species, and I get here these anatomical structures. And for example, you find some are very simple. So liver is called liver in both zebrafish and humans. So the homologous structure is liver, and it's homologous at the vertebrate level, meaning among all vertebrates, liver is homologous. Brain, for example, is homologous among all bilaterian animals. So basically all animals you can think of usually. But some are more tricky. For example, here we have lung in human. Now, zebrafishes don't have lungs. They breathe through the water through their gills, but the gills are not the homolog of the lung. The homolog of the lung in ray-fin fishes is the swim bladder, which is an organ which can take more air in and out to change their buoyancy in the water. So we here have lung and swim bladder, which are homologous, which are the homologous structure. So it tells you that the homolog of in human lung is the zebrafish swim bladder. And so if you wanted to compare the gene expression from GTEC to gene expression in zebrafish, this would be the comparable organ. And it also tells you which ones don't have homologs, either because there is no homolog or because we didn't yet manage to annotate it here. And thanks to the fact that we have the orthology of the genes and the homology of the organs, we can now compare expression between species. So here we have this tool called expression comparison. You put in a gene list, any genes you would like from different species or the same, but if they are different species, we'll use the homology of organs, and we'll give you all the organs where these genes are expressed in the different species. So here I put a gene list of genes which are supposed to be brain specific. This is you recognize on some IDs from human, mouse, chimpanzee, the other chimpanzee, the cow, and so on. And we find that, for example, 3 of these genes are expressed in the cerebral cortex in the different species, in the 3 species where they exist, and for 8 of the species I don't have data. This is because you have more or less data in different species. On the other hand, in the cerebellum, which is more studied, 9 of the species have data with expression. And I have the 9 genes are all expressed in the cerebellum. So you see that it's very conserved, the fact of being expressed in the cerebellum, in the metacephalon, in the hindbrain. And these specific structures are recognized as homologous because here I only have mammals. If, of course, I had more diverse species, I might not have the same level of detail of homology because you cannot find a homolog between the human metacephalon and the structure in the Drosophila, for example. So that depends on the level of species you're going to look at, taxonomy. Recently, we have added a new feature. So this is new to this course and to you because it was put online in Bgee last month. We have a gene expression matrix which allows us to more visually see these comparisons. So here you have different paralogs of olfactory receptors in fly, Drosophila melanogaster, and you see the anatomical structures where they are expressed. By default, it's folded, and the color code corresponds to the level of expression, the score which Frédéric showed you. And we have also our present absent code, which means that if it's absent, There is nothing. It's white. And gray means there is no data. So it's a difference between no data and there is data and we say it's absent. And what you see here is that these very specific olfactory receptors of Drosophila are present only in their specific olfactory receptor neuron. So each olfactory receptor neuron in Drosophila expresses one olfactory receptor. That's how olfaction works in Drosophila. And I know this because we know it from the literature, but we did not put this knowledge a priori in Bgee. We treated these data like all the others. They went through the same standardized annotation and processing pipeline. And this information, very specific information, was recovered at the end of this pipeline. So that you see that indeed, if I look in general at the sensory system, of course, all the olfactory receptors are expressed, but they're also each expressed in their specific olfactory neuron. And this, for now, has been implemented, released only within species. We will in the future release it between species. So to show you the same as the previous slide as this, but more graphical. And you can assess directly if the paralogs are annotated in the ontology database. Directly in the gene page, if you click on paralogs, you can access, you click instead of table, you click graph, and you will get to this view.
And sorry, Marc, but gray means not expressed and not.
Sorry. That's the opposite. Okay. White, no data. Gray, not expressed. Sorry. So this is a little WooClap, which is a bit different. It's more for us to know about you and to know what answers you need. So how do you expect to learn more about your favorite gene? So from the different tools you have in Bgee and the different views, what you saw this morning or this afternoon if you're in a different time zone, how do you expect to learn more about your favorite gene or genes? So some of you maybe study one specific gene or two, and some of you maybe a whole process or an organ. So I'll show the results here because there's no right or wrong. We don't have to answer expression comparison just because that's what I just spoke about. Interesting. So this is very interesting for us because it also tells us where we should put more efforts in developing tools, right? Because every one of those views and tools is months of work behind. Okay, so many of you are interested in the expression comparison. Quite a few in the present absent calls. So these are very much specificities of Bgee. And it's good to know that we're going to increase the number of species, which would make these more powerful. Some of you, the raw data annotations, and some topanat. So thank you for this. And so this concludes till we get to the final, and then we'll go to open Q&A about everything, about Bgee. So as I said this morning starting, I think you hope you see that what we want to do is to understand gene expression and then use our understanding to make gene expression useful to you. So that you don't have to worry which protocol this was, how do I normalize, was this cell type or that cell type that they meant, was this the correct annotation, am I here comparing really healthy wild type, or is this actually the brain of someone who died with Alzheimer's? All these questions should not be your concern because we take care of them so that gene expression is easy to use and can make you advance your science. So that's the end of my presentation. Stop sharing. There. So now there are new messages in the.
So there was a question I answered in the chat directly. How is it to obtain auto MCL? So we use OMA, and I put a link in the chat and document.
Thank you. Yeah.
Otherwise, yeah, we have like 3 generic questions, so don't hesitate to add more in the chat.
Are there questions? And if some of you want to put on your camera for this to say hi at the end, you're welcome. So some general questions. So, are you going to include plant model species at some point, for example, Arabidopsis? That is one of our most frequent questions, and I'm very sorry to say the answer is no. That is because the work on anatomy and cell types asks for a lot of expertise, and we have developed this expertise for animals, and for plants, it's a completely different expertise. So we're already a pretty big team, and we'd need to hire like 2 more people to have this expertise on plants. And so also because we are keeping up to date on the quality of genomes in animals and what are the projects and where they are stored and so on. And for plants, we would have to redo everything. So no, I'm sorry. Are you planning to include more species such as Sayona intestinalis? So as Frederick already wrote, we might include in the future. I don't know why there's a but in the sentence. So I have the list of species we're going to include so I can check it to tell you exactly. But we intend to, as I said at the beginning, to include all species of animals which have sufficient data in the next year or so. So let me just tell you. Well, the Sayona is in the list. So in the 200, which will be in the next release, there is Sayona intestinalis. So yes. Whether to consider TPM of fold change? Well, so Frederick answered. I don't know if you want to expand a bit on your answer.
Well, yeah, it depends on the question you're asking. So in Bgee, we look at gene expression per condition. So really, we want to report the genes expressed in a specific condition. We also want to make that comparable, but fold change is really about differential expression analysis, where you really compare two or more specific conditions and want to say with a statistical confidence, okay, it is twice as much expressed, 3 times more expressed. So, and usually, yeah, for doing the differential expression analysis, you're gonna use TPM, but some methods are based on read counts to do the differential expression analysis. So it's really 2 different questions. TPM is more like how much is my gene expressed, and fold change is really I want to compare specific conditions.
So the person asked about Sayona asked also about Spodoptera, and I do not find it in our list. Sorry. But I actually don't know what that species is. So I can look it up. But,
If you have any other question.
about Spodoptera, but also any others of you. If you know great data sets of gene expression that we might have missed because we try to follow as much as possible, but we don't manage to see all the papers and all the data sets published and so on, we have scripts to recover and tell us when new data sets come out, but sometimes they miss. Don't hesitate to contact us. You can just use the Bgee contact email on the website. It creates a ticket for our team. So if you see, okay, there's this data set which was published, which might be of interest, and I would like it, then send us. And because someone asked for it, we put it higher in the priority list. We need to know that there's data. So a good quality genome annotated, because we won't do the genome annotation ourselves, and gene expression of sufficient quality and a diversity of organs or cell types. Again, if someone, this moth, so I just checked, it's a moth. If this moth there's only whole body adult male and female, for example, or something like that, then we will not be able to do much with it because we focus on anatomy. If someone did a single cell atlas or dissected several individuals and has all the different organs, then yes.
Otherwise, I don't see any other questions. So you can still put questions if you want, but thank you for attending and your participation in the WOO claps and the chat.
Thank you very much, all of you. If you want to put on the cameras for a minute just to say bye. I see some molecular biology lab in the background, some offices. nice diversity. So thank you again for attending and for your participation, your questions, as Frederick said, and don't hesitate to reach out and we try to answer in a timely manner. And, all as resources, and I speak for the others here also, we don't have that many people who think reaching out to us. So when you do, we really take into account. I mean, or even you would think Uniproto Ensemble, we've reached out to them and they answer super fast. I've added also species to NCBI taxonomy. Don't hesitate to reach out to all your favorite resources to improve them. It improves us all. Okay? Thank you. Have a great day.